Okay, hello everybody. Good to see you all. Okay, so don't know about you, but I spent my weekend preparing later on lectures for this course and also watching and following the, well, I don't know what the right word is to describe it, the trials and tribulations of the Scotland football and rugby team over the weekend. Just if you get the feeling you want to talk to me about this, please don't, okay? Just it's a touchy subject. I need some time to process things. But we all believe everything's going to work for Scotland tonight, right? Definitely beating Denmark for sure. Hello. Before you come in, one question. Is Scotland going to beat Denmark tonight?
Yes.
Yes, exactly. Correct answer. Okay. Right. Um, So we're getting into the virology parts of this course. So just to start us off, anybody know the virus that killed Louis XV? Just on the off chance. No. Okay, he reigned for a long time. He caught this infection as an adult. It was very severe and he died. So it was smallpox. He was killed by smallpox. And at the time, well, okay, this is a bit of a random knowledge here, but at the time, people had very, very strange ideas how to treat this kind of infection. So there was a big thing was the red treatment. You had to like cover or surround people with the color red, and that was going to help you. But it did not help at all, so he was killed by this. Okay, the next viral pandemic could be, any ideas on this one? Okay, what was the last viral pandemic? What was the last one? Yeah, COVID-19. So it was a coronavirus was the last one. So the next one could be, well, it might be another coronavirus. Who knows? And previous ones, what have they been? Huh? Oh, unlikely for Ebola virus to become a pandemic. Why? Because everybody is too sick. When you get Ebola hemorrhagic fever, then you are really, really sick and you don't move about very much and you can't transmit it to so many people. So personally, I doubt that, and there's no real like airborne transmission. It has to be direct transmission with the person. So it's not the best candidate for like massive pandemic that can infect millions of people, fortunately. But previous ones, huh? Smallpox has been eradicated. Oh, it could escape, you think?
No, I saw you ask previous.
Previous pandemic. Well, I don't know. This is a question. Was smallpox a previous pandemic? Probably at some point, but we don't know so much when it emerged in the human population. It was a massive, historically important pathogen in the colonization of the Americas because the American continents were free of smallpox until European colonists arrived. And it really had a huge impact on the populations of the Aztecs and the Incas. It's a big factor in how Cortez and Pizarro were able to like bring down these big organized societies because they had like measles and smallpox virus arriving at the same time. Okay, but yeah. Yeah, maybe like influenza virus. Yeah. So Spanish flu, and there have also been other pandemic influenza between since 1918, 1957 and 1968. And also like 2009, the Mexican flu, but it was not very pathogenic. But it was a new virus that infected millions and millions of people worldwide. Okay, so probably, if you had to put some money on it, might be another influenza virus or another coronavirus. Okay, right. So that's just kind of the warm up. So what we're going to be doing is mostly I'm going to be presenting fundamental virology from a molecular point of view. Okay, so what we're doing today is have a closer look at the structure and classification of viruses. And then we'll have 3 lectures on the replication cycle of viruses. Firstly, to look at attachment and entry, mechanisms of replication expression, and then at the end, how the viruses get out of the infected cells. And then afterwards, we'll talk about virus pathologies and plant viruses. Probably I'm going to switch around the order. I will probably do plant viruses first, because in the past, when I've done this course as a kind of option, I would talk too much about this and I didn't leave enough time for plant viruses. So I'll switch them around so that at least I won't run over the time. So that's our program. And so for virus structure, so just a few reminders from like 2 years ago, maybe. So just remember most viruses, the size of the infectious particle is less than 200 nanometers diameter. So that was one of the first ways to distinguish viruses from bacteria. You could filter, well, viruses were filterable. That is, if you passed the infectious material through a filter, bacteria will be retained on the filter, viruses would pass through. So how do we know anything about virus structure? Because they're so small, you can't see them in an optical microscope. It's not like gram-positive, gram-negative bacteria. So you have to have powerful techniques to visualize their structures. So to see a whole virus particle, this became possible with the development of electron microscopy, and specifically electron microscopy or transmission electron microscopy with negative staining. So this was invented in the 1930s. We got the first pictures of viruses in about the 1940s, and notably bacteriophages like this one. Now you notice the background is dark and the phage particle is kind of white here. That's negative staining. Do you know about how this works from like other classes? No? Okay, well, I'll just talk a little bit about this then. Years ago, I had some slides on this, but I couldn't find them anywhere. Okay, so the way electron microscopy, negative transmission electron microscopy works like this. You get your sample, like imagine you've got a virus particle like this. You have to adsorb it onto some kind of like metal platform or kind of like a, the sample grid. And then you have your electron source that is blasts through it, okay? And you have the detector down here. And this is in a vacuum, okay? So you've got to adsorb the sample to the sample grid, remove all the air, and then you blast it with the electrons. Now the problem with this is that biological material, proteins, lipids, they are transparent mostly to electrons. So if you just put your sample on there and you look at the photographic plate underneath, probably the electrons all get transferred. So what you do is you coat, after you've adsorbed the sample, you'll treat the grid with uranyl acetate. Okay, so you have a heavy metal ion like uranium, and this is going to deposit all the way around here where you don't have any proteins. Okay, so you've got a lot of big heavy metals there, and they will block the electrons. So this becomes like impassable, the electrons can't get past, and you only get the transmission where you've got some like biological material. So that's why you see something like this, okay? All the black parts, this is where you've got the uranium ions that are on the grid, and they're blocking the electrons. That's why it's black. And where it's when the electrons are transmitted, that's where you've got the protein. Okay, so uranyl acetate was big classical contrast reagent for transmission electron microscopy. Now there are like other alternatives that don't have such dangerous metals. But that's how you get this. Now, another or more advanced type of electron microscopy is cryo-electron microscopy. Now, one of the disadvantages with transmission electron microscopy is that the physical conditions to prepare the sample can be quite rigorous. So the sample has got to be dehydrated and put under a vacuum. Now that works pretty well with some non-envelope virus particles, but it can cause damage to more fragile viruses. So in order to get an accurate vision of this type of particle, you use cryo-electron microscopy. So in this type of microscopy, you'll have the viruses will be embedded in a thin film of, well, aqueous solution, and this is flash frozen, so you get a frozen vitreous ice here. Okay, and then this whole thing goes into the sample grid, and then you get the electrons passing through here to get the image. So the advantages are that the particle stays in an aqueous environment. There's less risk of damaging the structure by the sample preparation. So you get a more accurate image of the real particle. And when you have that, you can get something like this, okay? Now, as I said, the problem is that proteins are mostly transparent to electrons, but with advances in technology, you can get a little bit of contrast. So you can see the background is light gray and the particle is dark gray. Okay, the signal's weak, but you can get information out of it. And what you can also do with this is this whole thing can be tilted, and you can get some information about the three-dimensional organization of each individual particle. So when you see an image like this, and you get data from this kind of preparation, it allows you to have an idea of the three-dimensional structure inside and outside the particle. Okay, so that's cryo-electron or microscopy or cryo-electron tomography. Really, really important and getting more so important structural technique in virology. And as electron microscopes get more and more powerful and we have better computational tools for the data, some of them you can get a resolution up to atomic. Okay, you can see the single atom level structures. So that's possible for some viruses. Mostly atomic level resolution is given by X-ray crystallography, either for whole viruses or virus proteins. So to get a crystal structure of a whole virus particle, have you seen something like this in protein biochemistry? You need to get the crystal, right? And to get the crystal, every single particle has to be identical. So that can work for non-envelope viruses, but for envelope viruses generally doesn't work. Okay, so X-ray crystallography, you can get the atomic structure of viral proteins and sometimes the whole non-envelope virus. Okay, so that's how we know about virus structures. Okay, we've got these kind of techniques. And once you apply them, what do you see? Well, we can see that like the smallest type of virus particles, maybe the minimum are these like small kind of specks with maybe less than 20 nanometers diameter. And this kind of simple virus particle is made up of 60 copies of one protein. And inside you'll have the virus genome, and this is perhaps the smallest example I could find, just 1,762 nucleotides of single-stranded circular DNA. Codes for 2 genes. One gene codes for this protein, the capsid to make the particle, and the second gene will code for a protein that allows the genome to be replicated. And that is the absolute minimum for a virus. One gene for the capsid, one gene to promote the replication of the virus genome. That's the absolute minimum. And at the other extreme, you've got these giant viruses like the Pandora virus with a massive looking particle like this. It's so big you can see it by light microscopy. These were originally thought to be intracellular bacteria, but no one could actually culture them. So for years and years, it was a big mystery. And I must say, this is the major contribution of Didier Raoult to microbiology, I think, was his like first description of these giant viruses. Anyway, so Pandora virus is really the biggest one that we found so far. The genome is 2.5 million base pairs of double-stranded DNA. That's a bigger genome than many bacteria, than some bacteria. Coding for about 2,500 genes, and inside this particle, there's more than 100 different proteins in there. So it's a very large, complicated structure. However, most viruses are situated here, okay, less than 200 nanometers in diameter. Oh, I got this wrong. What's going on here? Okay, so based on electron microscopy, the two different types of virus particle are called enveloped and non-enveloped. So non-enveloped or naked viruses are constituted just of the genome and the protein code called the capsid. And enveloped viruses, they've got the same kind of thing on the inside, a nucleocapsid, and then a lipid bilayer on the outside, which is called the envelope. Now, among non-enveloped viruses, there's basically two different types of capsid structure. There are kind of like long, elongated, stick-like structures with helical symmetry, or there are spherical structures with icosahedral symmetry. And bacteriophage, for example, they have a kind of association of these two different elements of structure. So you've got, like on a bacteriophage, you'll have an icosahedral head and a helical tail. Now, amongst envelope viruses, I mean, you could say that simple envelope viruses, they've got a nucleocapsid, which is either helical or icosahedral. So for this one, vesicular stomatitis virus. Okay, I'll tell you why all my letters are like this. It's really annoying. Okay, so I work with LibreOffice Impress, but if I have to change it to PowerPoint format, it always like does this to every single text box, and then you have to like fix them one by one. So that's why it's like this. Okay, please excuse me. I didn't have time to go back and fix them all. So, okay, so vesicular stomatitis virus, you can see it's got an elongated nucleocapsid with a helical symmetry. Whereas this one, this is cytomegalovirus, you can see you've got an icosahedral capsid just like this one inside, and then you've got an envelope around it. And next to that, we have the Pandora virus. Doesn't really correspond to either one of these two situations. So we just call them complex, okay? Complex structure. The other thing you need to know about electron microscopy is many virus families were named because of their morphology, okay? So coronaviruses, this is the crown of the spike glycoprotein around the particle. Kediciviridae looks like a kind of cup on the surface of the particle. And arenaviruses, okay, they don't like have gladiators fighting or anything, but arena means sand. And so the inside of the particle had a granular kind of, well, look or granular quality to it. Okay, so structures are very, very simple, okay? Helical, icosahedral, envelope, non-envelope. We've got like 4 categories, very simple. And we don't have very many proteins, so it's going to be easy to understand what the structures, what the functions are of each of these different elements of the structure. So the first thing is the capsid or the nucleocapsid. The primordial function of the capsid is to protect the virus genome. Because for every virus, at some point, it's got to leave an infected cell and find another cell to infect, or another organism. And if you just leave a free molecule of RNA or DNA floating around in the sea or circulating around in your blood, it's not going to last all that long, okay? It's going to get degraded. So having a capsid is essential for infectivity. And the main thing, protecting the virus genome. Now, also for non-envelope viruses, the function of the capsid proteins are to bind to and allow the virus to infect a new host cell. So they have a function of attachment and entry. Now for envelope viruses, okay, you've got the capsid like this, and you have the envelope like that. Of course, and at some point, the virus is going to need to bind to a host cell with a receptor molecule. The capsid proteins, they're on the inside now, so they can't interact with this receptor. So every envelope virus has to have at least one glycoprotein that can interact specifically with the receptor. So every envelope virus has at least one envelope glycoprotein, and its function is to attach to the entry receptor and allow the virus to infect a new cell. Okay, so that's kind of easy. And the other thing you can see in, yeah. Can a naked virus have a glycoprotein? That's a more complicated question than you think. Okay, where do proteins get glycosylated in the cell? Because that's where the answer is going to come from. Yeah. Yeah, so you've got the secretory pathway, endoplasmic reticulum, Golgi, and then secreted. So that's where glycosylation happens. So if a non-envelope virus is going to have glycosylation, it must go out through these compartments. Okay? Let me draw a fantastic looking cell around here. That's the nucleus. And then, I don't know, we've got the endoplasmic reticulum, the Golgi, something like that. And then secretion. Now imagine you are a non-envelope virus, naked virus. You're producing your particles here, and then they get released by cell lysis. Can this get glycosylated? No. If you're producing your particles in the nucleus, and then they get released by glycosylation, or by lysis, can they get glycosylated? No. So the answer to your question is most non-envelope viruses, their capsid proteins are not glycosylated. But there are some exceptions. So rotaviruses that cause gastroenteritis, they actually have a complex secretion pathway, and they do get released here, and then they have some of their capsid proteins can be glycosylated. But it's an exception. Most of the time, the particles are formed in the cytosol or in the nucleus, and they cannot get glycosylated. Okay, that was a bit of a lucky question for me because it was complicated, but it's one of the, I have a good idea about the answer. Yes, please.
So it's possible for non-enveloped virus to become enveloped?
That's not the same question, I think. I think what we're talking about here is whether on the capsid proteins you have some glycan structures, okay? Okay, and whether some non-envelope viruses can get a kind of lipid membrane. We'll talk about that when we do assembly and release. Okay. Right, so I was just going to talk about these. Okay, so you've got the envelope, you have the capsid, and in between the two, there are often some other proteins, which are called the matrix or the tegument proteins. So the matrix proteins, like here, it's this kind of white blocks in between the nucleocapsid and the envelope. They have a structural role. They just stick in the whole thing together. And they also play an important role in the morphogenesis and the release of envelope viruses. For herpes viruses, there's this kind of like more amorphous region within the particle, which is all this like orange section here. And this has several different proteins, which are called tegument proteins. Now, they don't really play a structural role. Well, maybe some of them do. But more importantly, what their function is that they can be injected into the host cell right at the beginning of the infectious process. So this is the host cell. This is my envelope virus particle. And if I've got some tegument proteins here, like this, the way this virus is going to infect the cell is to stimulate membrane fusion. So what's going to happen is that the virus envelope and the cell membrane here are going to fuse together, and this means we have a pore going here. So everything that is inside the virus particle can now get into the cell cytoplasm. So you could have like the whole capsid, and it will do something else inside the cell, but you also have these tegument proteins. And these tegument proteins can go around and have different functions in the infected cell and start to subvert the infected cell and turn it into a factory for producing viruses before we even get any gene expression from the genome. Yes? No, no tegument protein in a non-envelope virus. Only envelope viruses. Tegument and matrix is in between the capsid and the envelope. Yes?
Does that mean that envelope viruses are more efficient than naked viruses?
Are envelope viruses more efficient? Well, more complex. They're more complex. So they can do bigger variety of things. More efficient. Don't know. Yeah, I'll be a link later on. We'll come back to this question. Okay, so this has taken a long time, thanks to all your interactions, but let's have a question. We can maybe just do this by show of hands, right? Since it's not too many people. So just think about it and have a chat with your neighbor if you're not sure about an answer. Oula. Okay, everybody ready to express an opinion? Okay, so question one, you can remove virus particles from a solution by filtering it. Who says yes? Who says no? Yeah, you're right, they pass through the filter. Correct. Two, can you determine the structure of an envelope virus by X-ray crystallography? Who says yes? Who says no? The answer is no. To get a crystal, every single particle has to be identical. This can be true for naked viruses, but for envelope viruses, the envelope is too fluid. Okay, it's a little bit mobile. So two particles are generally not identical. So crystallography won't work with envelope viruses. So the answer is no here, because they don't form a crystal. That's it. You can concentrate them as much as you like. You'll never get a well-organized crystal. You might get some kind of like precipitate, but that won't work for crystallography. Let's have question 4 first. Some viral particles are larger than some bacteria. Who says yes? Yeah, you're right. Like the giant viruses can be larger than the smallest bacteria. So there's some overlap, okay? Okay, how about this one? Naked viruses are more resistant to detergents than envelope viruses. Who says yes? One person. Who says no? Many people. Okay, somebody who says no. Why did you say no? Yeah.
Maybe so, but for me, it's envelope. They have an additional layer of protection.
Ah.
So for me, the detergent will firstly destroy or create all, I don't know, in the envelope, and after it's destroyed, it can go inside.
Ah, okay. So because you have an extra layer, it should be more resistant. Okay, that's argument for no. Yes, what's the argument for yes?
Detergent will react with the glycoprotein.
Okay, detergent is important here because the detergent is like soap, right? It will dissolve the envelope, the lipid envelope. So with a detergent treatment, you take off the envelope. Will this capsid be infectious? No, because envelope viruses, they require the glycoprotein to infect a cell. They've gone, you've removed them. Whereas like the capsids will remain resistant to the detergent, okay? So this will still exist, but it will not be infectious. Non-envelope virus, well, you just cleaned it up a little bit, be shiny, and being sparkling, just like your dishes when you get them out of the dishwasher. Okay, so this maybe relates to the question, are envelope viruses more efficient than non-envelope viruses? Because this is a generally true, okay? Naked non-envelope viruses are generally more resistant to chemical stress than envelope viruses. So more resistant to drying out, desiccation, more resistant to detergents, yeah, maybe some other types of chemical treatment. So on a practical level, it determines, or to a large part determines, what kind of chemical treatment will be required to disinfect a surface. Okay, influenza, coronaviruses, they're envelope viruses, just washing your hands with soap will be good. Washing down the surface with a simple soapy solution will be okay. Noroviruses, which cause gastroenteritis, they're non-envelope viruses. You need to treat them with bleach to inactivate them chemically and leave it for a longer time. Okay, so non-envelope naked viruses are more resistant to chemical attack. Yeah.
Does hydroalcoholic gel work for to wash hands for naked virus?
Yeah, yeah, but probably less effectively. But I would have to look it up specifically. I don't really know that. But so, different types of disinfectants will have different activity against different types of viruses. And the envelope viruses will be the most sensitive. So in terms of animal infections, where do you have an environment that chemically is going to be very, very difficult for an envelope virus in your body? Okay. Yeah, in the gut, because you have low pH, and then afterwards, what do you get in the next part of digestion? Go to the duodenum, and what happens? Oh, you forgot all your basic physiology. What is the matter with you? Yeah.
The bill will.
Yeah, bile salts. Okay, so it neutralizes the pH, becomes alkaline, but also attacks membranes, right? So all the membranes will tend to precipitate. So most viruses that cause gastroenteritis are non-enveloped. Okay, so enteroviruses, astroviruses, norovirus, rotaviruses, non-envelope viruses. Only one exception, some types of coronavirus. And I don't know why that is. I really don't know. But I think there's something specific about their envelope structure that allows them to resist this chemical attack. So if you want to be more efficient at infecting someone's gut, you better be a non-enveloped naked virus, okay? So more efficient, less efficient, depends on what you need to do, okay? Okay, so let's have a closer look at virus structure, and I'm a little bit scared that we're going to go way over time, so I will try and be a little bit fast. So the basic idea of the capsid protein is that the job is to protect the genome. And genomes are nucleic acid, DNA or RNA for viruses, and nucleic acids are linear polymers. So one way to protect this linear polymer from getting degraded would be to get a load of proteins that form a ring and then carry on forming a ring around, so it forms a helix that goes all the way around this nucleic acid. And you keep on adding subunits to the end until the whole thing is covered. That's it, job done. You've protected your nucleic acid. And this is very, very simple architecture, which you find in a lot of plant viruses, like tobacco mosaic virus. It's got an RNA genome. The capsid looks like this, a rigid stick. And it's, maybe 1,000-ish copies of a single protein. And all these proteins wind around the nucleic acid, and that's it, that's the particle. So very, very simple, you protect the nucleic acid with one protein. So this kind of architecture is very frequent in plant viruses. In animal viruses, there are a lot of viruses with helical nucleocapsids, but they are all enveloped, okay? There's no naked helical viruses naked viruses with helical symmetry in animals. So you can see this with like the filoviruses, like Ebola virus. You can see the capsid, the infectious particle is like, rod-shaped. But also you get some viruses like measles virus where the capsid is kind of filamentous and flexible. And it gets kind of rolled up into this envelope particle. Okay, so helical nucleocapsid, very, very simple idea, but there's a problem with it. And the problem is that once you get to a particle size like this is about 15 nanometers in diameter, but it's probably already about 200 nanometers, 300 nanometers long. And the longer this type of particle gets, the more fragile it becomes. It can actually, snap apart. And that's going to impose an upper limit on the size of the genomes that viruses can have. If their particle is made like this, once you get past, 5, 6 kilobases, 7 or 8, then it becomes difficult to maintain a stable particle. So there's two solutions that viruses found. One of them is to distribute the genome over multiple segments. So once again, some plant viruses do this. If you purify virus, then you'll get particles that have different sizes. So these long ones, they've got a long genome segment, and the short ones have a short genome segment. But to infect a cell, you need at least one of these particles and at least one of these particles to infect the same cell at the same time, which also causes its own problems. But if you do that, then you can kind of like overcome this barrier on the genome size. So you can have, both particles are stable, so you can overall have about 10 kilobases of genetic information. So the other way around this is to make the nucleocapsid flexible and then fold it up inside the envelope. So that's what you have in measles virus and the coronaviruses. They both have helical capsids, but they're flexible. They're not rigid. And because they get packed up in the spherical particle, the overall particle size remains quite small and stable. And that's also the basic idea behind icosahedral capsid. So the idea here is that the nucleic acid, we're not going to stretch it out in basically one dimension on a line. We're going to wrap it up in 3 dimensions like a ball of wool, and then it can be more compact. And then the capsid is going to form a spherical structure around the ball of nucleic acid. Okay, so every icosahedral capsid is kind of like this. You've got a kind of shell of protein, and on the inside here you have the genome, the nucleic acid. Now the simplest way to do this is if you've got 60 copies of one protein. That's what you have in the PCV, porcine circovirus capsids, that were the simplest example. But these particles will be limited in size to 20 or 30 nanometers diameter. So if you want to get a bigger capsid, you've got to have a more complicated architecture. Okay, so now we have to talk a little bit about, geometry and geometrical forms. So the regular solids that are roughly spherical are icosahedrons and dodecahedrons. So an icosahedron is made of 20 triangular faces, and a dodecahedron has 12 pentagonal faces. And both of these have the same axis of symmetry. So as you see them going round, you can see that each vertex on the structure here has got like three, what's the word I'm looking for? Edges coming from it. It would be arête in French. So you've got a threefold axis of symmetry coming from each vertex here. Then at each edge, you have a twofold axis of symmetry, and in the middle of each pentagon, you have a fivefold axis of symmetry. If you look at the icosahedron, the threefold axis of symmetry is in the middle of the triangle, and the fivefold axis of symmetry is at the vertex, okay, because you've got 5 edges coming from each vertex. So they both have like 5, 3, 2 symmetry. So they're kind of equivalent. Now, what does this mean for virus particles? So the simplest case is if you put together 12 pentagons. Okay, if you have a pentagon, 5 copies of one protein, 12 times 5, 60 copies. It's the same if your protein makes up a triangular face. 20 faces, 60 copies of proteins, of the protein. Now, if you want to get a bigger particle, what you can do is you can add some hexagons in there. Okay, so you've got the pentagons, and each pentagon will, in the center of it, will be at the vertex of the icosahedron. And it's also true if you put in some more hexagons. And you can keep on doing this. So you can add even more hexagons and even more hexagons. Right? And then you'll get a bigger and bigger particle. And every time you do this, you've always got exactly 12 pentagons and a different number of hexagons. And the pentagons, if you connect them, this always gives you a triangular face. Okay, that's why all of these kind of shapes have icosahedral symmetry, because you've got 20 of these triangular faces. Yeah.
I want to understand where the 12 pentagon.
Okay, it comes from, what is a dodecahedron? 12 pentagons. This is the simplest case. So you start with 12, it's a dodecahedron. But if you join up the center of all the faces of the pentagons, that's where the triangles come from. And if you add more hexagons in there, the thing gets bigger and bigger, but the pentagons are always in the same relative position on the surface here. Okay. So that's the general idea. You can make a structure that's spherical, and you've got to have a mix of pentagons and hexagons. Okay. Now, the reason for this is because, okay, what happens if you've got no pentagons? Yeah. Why the hexagon? So it can get bigger. You put more in. You see this one? We put in some extra hexagons. So these guys, they could probably all fit together, right? But if they go together, it's going to be small. You put in a ring like this of hexagons, the whole thing can get bigger. No, you don't see this? You can't see that this is bigger than this? Okay, well, this is one of these things. I think you're going to have to talk about it amongst yourselves, because I don't know how to explain this any clearer. This is going to be I don't know, honestly, because Okay, another way to think about this, and the reason structural biologists began to think about this in the 1960s. This was actually Francis Crick who pointed out this problem in virus particles. He said the genome is very small, and a particle is made out of protein. It's got to be it will have to give you a structure of different size. Okay, so the average size of a virus capsid protein might be something like, I don't know, 300 amino acids. And this will give you 1,500 base pairs of DNA to code for this. Okay? Right. So imagine you're going to make your particle, it's got 60 copies of one protein. You just need this amount of size in your genome to do this. Now, if you want to make a big particle this size, it's going to be 10 times bigger than this one. But you just make it with one protein. Then your protein will have to be 3,000 amino acids to cover the whole surface. That's going to give you 15,000 base pairs just to make one massive capsid protein. Imagine we want to get bigger. We just have a big pentagon, right? Really big. Every single protein will also have to get much bigger. And then your genome is too big. Can't work. So the solution is you keep one small protein. That makes this part. And you get maybe another small protein, will be the hexamer. Will also be like 300 amino acids. And that will give you 1,500. But you put this together, you've got three kilobase of DNA. You can fit that whole genome in there, okay? Okay, really sorry, that's maybe not helping things, but that's the reason why viruses do this. So, you know, it's like if you want to get a bigger football, add some more hexagons, the whole football will get bigger, honestly. And this kind of structure you see everywhere. You see it in a football, Buckminster Fullerene, this kind of like a geodesic dome. Mostly you look around, it's got hexagons, but from time to time you will see a pentagon up here, like this one. And you need to have the pentagon because that's what I was going to say before. If you just have hexagons, what structure do you get? Nobody plays board games here with hexes? Okay, so I'm going to get a completely flat surface. How can I turn this into a three-dimensional object? Well, maybe I could fold it round into some kind of tube, right? It would like fold round. For these ends, it would always be open. I would never be able to close the end, just if I have hexagons. So in order to get the curvature like this, I need to have the pentagons in there. So that's why, whatever, you've always got 12 pentagons and a different number of hexagons. And people in Imperial China, they knew this. If you go to the Forbidden City, you can see actually this statue. And it's got a load of hexagons here, and from time to time they put a pentagon in there because they realize if we don't, it's never going to like close around. Okay, so with this idea, we can kind of, if we get the capsid and we flatten it out on the surface, what we'll have is we'll have a hex lattice, but from time to time, instead of having a hexagon, we'll have a pentagon. And people who do structural biology, they thought, okay, so we need to like have a formal way to explain where these pentagons are with relation to each other. So there's two axes in this lattice here. There's one k-axis and this one is the h-axis. And the basic idea is, how do we get from here to here? You have to count how many steps you go in one axis. So I go 1, 2, 3, 4 steps on the h-axis, and then 2 steps on the k-axis. And it would be the same if I went the other way around. 1, and then 1, 2, 3, 4. Okay? So we've got h equals 4, k equals 2. So the crystallographers, they'd want to combine this into one number, which is called the T number, or the triangulation number. And it's calculated like this. It's h squared plus hk plus k squared. So in this example, it will be 16 plus 8 plus 4, which gives you 28. Now this is a number which characterizes an icosahedral capsid, okay? And it summarizes the relative positions of the hexamers and the pentamers. So, and it's something that's in all the databases to say, okay, this type of virus has got a T equals something icosahedral capsid. So how about this one? What's the T number of this one? Just thinking about this. Everybody got their eye on it? Everybody, can everybody see the pentagons here? Maybe not. Okay, this is a pentagon, 1, 2, 3, 4, 5, and this is a pentagon, 1, 2, 3, 4, 5. Everybody ready to put your hand up? Who says my T number is 4? Oh, before this. Let's do it. Or rather, okay, what's my H number going to be for this one? What's the K number going to be? So this would be, it's not on the list. Yeah, it's 4. So we go from here, 1, 2, 3, 4. 4 steps to get from here to there. So it's 1, 2, 3, 4, 16. Now, what about this one? So it's difficult to see where the pentamers are, so I've put them in red here. So what's the T number of this one? Oh, damn it. Okay. This is kind of here, it's on this one, not on that one. If I go 2, 1, I can go 1, 2, but I can't, 2 doesn't get me there. I need to go 1 and then 1, 2. So it's 4 plus 2 plus 1, T equals 7. Okay, so that's what these T numbers mean, okay? And that's how you calculate them. So the basic idea is that the bigger the T number, the bigger the particle diameter is, okay? You got more hexagons in amongst the pentagons, bigger T number. And basically, the bigger the particle is, the bigger the genome can be. Now, what's happening here? Okay, you've got picornaviruses, the particle is 27 to 30 nanometers diameter, and the genome is 7 to 8.5 kilobases long. Hepatitis B virus, slightly bigger particle, smaller genome. What is happening here? Oh, somebody else. Anybody? The answer's on the slide. Yeah. So I got double-stranded and single-stranded. Okay, so this takes twice as much space. So if you take into account double single-stranded, then the relationship between size of the particle and the amount of nucleic acid inside it holds. Okay. Okay, so up to now we've considered these hexagons and pentagons as a kind of abstract mathematical form, but they're made of proteins. So often what you have is that one capsid protein will form the pentamer, and then maybe one or two others will form the hexamer. So that's what you have in picornavirus capsid. So VP1 forms the pentamer, and VP2, VP3 form the hexamer. But also you get the situation where it's the same capsid protein which can form a pentamer or a hexamer. So in herpes viruses, that's what you have. The pentamer and the hexamer, it's the same protein, but 5 copies or 6 copies. Okay, so special case of lentiviral capsids. So these are made up of the capsid protein, CA. And if you look at the shape of it, you can see that it's a kind of like cone-shaped. It's not really spherical. And the way it works is you've got mostly hexamers, but you've got 5 pentamers down here and 7 up there. So you have to have 12 in order for the whole thing to close the curvature. So it's a kind of icosahedral capsid, even though it doesn't look like one, okay? And that's in lentiviruses. Other retroviruses have got a kind of regulus-shaped capsid like this. So if you've got something like that, that's lentivirus. This is another type of retrovirus. But it's basically, this is just a kind of variant. Okay, so these capsid proteins can form pentamers or hexamers. And one thing that was discovered, my goodness, 40 years ago now, is that viral capsid proteins have a specific kind of structural fold. So the beta barrel jelly roll is found in virus proteins and in some cellular proteins, but the viral ones are rather different. And so they're very specific to viruses. So if you see this kind of protein sequence in any kind of like, DNA sequencing project, you know you've got a virus somewhere. So the beta barrel jelly roll fold is what's called a virus hallmark gene. Only viruses have this. And it's got, what's good about it is that it forms this kind of like trapezoid structure, which gives you a kind of brick. And then you put 5 bricks together, it will give you a pentamer. 6 bricks together, it will give you a hexamer. And you put them together, that's how you get this capsid. Okay, envelope viruses very, very quickly. So there are 2 kind of types of viral envelopes. Mostly the envelopes are fluid. So the lipid membrane's fluid and the glycoproteins are kind of diffusing around inside the envelope. But there are envelope viruses that have got a rigid structure. So, dengue fever virus, this is a kind of section through the particle by cryo-electron microscopy. You've got the virus RNA in here. And then you've got the lipid membrane. You can see the 2 parts of the bilayer, and then the protein coat around the outside is highly organized. So it's a rigid structure, pretty much like a capsid. Now, what about envelope glycoproteins? What are their functions? The main functions are attachment and entry. So every envelope virus will either have one or two proteins that are performing these functions. But you can also have some other things. Okay, sometimes you've got viroporins, so these pore-forming proteins in the virus envelope. And you can have some other envelope glycoproteins doing other things. Now, all envelope virus proteins have to have a fusion protein or a fusion domain. And fortunately for us, there are only 3 types of fusion glycoproteins. So the type 1, they are trimers of alpha helices that stand up perpendicular to the virus membrane, virus envelope. So influenza, HIV, SARS-CoV-2, they're all based on the same principle. Type 2, fusion domains, fusion proteins, they're formed of beta sheets, and their basic structural subunit is a dimer, and they're flat on the surface of the virus. So that's these ones here. Okay, they're lying down flat on the envelope. And then other viruses like some herpes viruses and rabies, they have a type 3 fusion domains, kind of a mix of alpha helices and beta sheets, is standing up, but this structure very, very different from this one. Okay, so it's a different class, but they all do the same thing. Okay, so I've got maybe 15 minutes to talk about classification. So in the last, certainly in the last 10 years, there's been a huge update of virus classification. And that's because we've had a big, influx of millions and probably billions of base pairs of high throughput DNA sequencing data. So if we go back 40 years ago, if you wanted to describe a new virus, you have to isolate it, culture it, purify it, and then you get the genome. So it's slow process. Now, if you want to get a new virus genome sequence, take a bit of water out from the air, extract the DNA and sequence it. You probably get 30 or 40 new viruses, something like that. So there's this huge amount of new sequencing data, and the idea is how are we going to identify viruses and classify them? So the ICTV, the International Committee on the Taxonomy of Viruses, had to really make a big effort to get a kind of structure where they could handle all these new sequences and try and classify them. So the idea is that now virus classification is really dependent on molecular sequence homology. And basically, you can try and define bigger and bigger groups. So now if I just go through this, and the idea is we're going to try and make virus classification consistent with the rest of the classification in biology. So we'll try and have the Linnaean type divisions here: kingdom, phylum, class, order, family, and then have a higher one up here, which is called the realm. And the word endings here are telling you what level you are in the hierarchy. So Viridae is a family name, or Viridae, I suppose you say in English. Viral is the order level. Viricetes is a class level. Viricotta is a phylum level. And if you got something virus all in one word, that's a genus level. There's a big effort to try and have new names for viruses which have a binomial structure. So the first word in the name is going to be the genus name, and then the second specific for the species, to make things consistent with the rest of biology. So the idea is that we can group together all the RNA viruses in this realm of riboviria by, for example, the molecular phylogeny of the RNA-dependent RNA polymerase. All RNA viruses have got to replicate their genome with an RNA polymerase. And no cells do this. Transcribe RNA to RNA. Doesn't happen. So this is a specific virus gene. It's conserved in all RNA viruses. So it's a homologous character you could use for phylogeny. So this is what allows us to kind of class the negative strand RNA viruses up here, and these are double-strand RNA viruses, and these are all the single-strand RNA viruses, positive strand RNA viruses. So it gives you an idea of what the ancestors were. The ancestors were plus-strand RNA virus, then some of them became double-stranded, and some of these became negative-stranded. Okay, so that's all very nice, but you know, a lot of virologists moan about this. Why? Because the names have been changing a lot. So the old name, like people like me, we learned these names. You would have like respiratory syncytial virus, Epstein-Barr virus, and in plant virology, things like maize-streak virus. So where do these names come from? Well, the people who discovered it, okay, Epstein and Barr, the discoverer named virus, something to do with the symptoms or how you catch it. Respiratory, and it forms syncytia in cell culture. Maize-streak virus, the host plant, and something to do with the symptoms. So this is the kind of old names that everybody like me learned. And then more recently, there was a kind of first attempt to update. So everybody started learning, okay, Epstein-Barr virus is not called EBV anymore, it's called HHV4. And then it got changed to H-gamma herpes 4, human gamma herpes 4. So now we've got like the new names. So all of these new names, like a lot of working hospital virologists or people working out in the field, plant virologists, they don't know these new names. Okay, the committee just telling us that they're going to be called this. So if you know, if you're like calling a clinician and you're telling them, oh yeah, we detected orthopneumovirus hominis in your patient, they don't know what you're talking about. Okay, everybody still uses the old names here. And it's actually very difficult for me to like find out, well, what was actually this one? Oh, it's the RSV. So that's the ICTV classification. The other classification that kind of still works in parallel is what's called the Baltimore classification. First proposed by David Baltimore at the end of the 1960s. He just died last month or the month before. Real significant figure in 20th century biology. So David Baltimore, what he said was, okay, in order to understand how viruses replicate, we shouldn't classify them by whether they give us a respiratory infection or a gut infection, which is what clinical virologists were doing. We need to classify them by the structure of their genome. So the first thing is we'll make a distinction between DNA viruses and RNA viruses. And then amongst the DNA viruses, I'll divide them up into viruses with a double-stranded DNA genome and viruses with a single-stranded DNA genome. Then for RNA viruses, I'll do the same thing. Class 3 is the double-stranded RNA viruses. But for the single-stranded RNA viruses, he made the distinction between plus-strand viruses, so the virus genome codes directly for a protein, and the negative strand viruses, so the virus genome is the complementary strand. So this has a big impact on how these viruses can replicate. So initially, that was it. He had 5 classes. He thought he'd covered all the possible situations. And that was true until the discovery of the reverse transcriptase, the discovery of retroviruses. So retroviruses have an RNA genome, but when they replicate, they copy the RNA into DNA via the reverse transcriptase. So this was a big, important discovery because it was the first, it was breaking the rules. It was breaking the fundamental dogma of molecular biology, which is that biological information goes from DNA to RNA to protein, one directional. But here you've got information transferred from RNA to DNA. It's going the wrong way round. This was a big surprise, got the Nobel Prize. Who won the Nobel Prize for the reverse transcriptase? This guy, and Howard Timmin. Okay, and viruses like hepatitis B also do the same kind of thing. They have an RNA intermediate in infected cells, but in the particle, the genome is DNA. So often these two, they're grouped together as the reverse transcripting viruses, because at some point they've got the information in RNA has got to be copied back to DNA. So the Baltimore classification, much simpler, related to virus genomes and how they're going to replicate their genomes. So it's very useful for understanding replication cycles. So people know the Baltimore classification. Now, one of the things you have to remember is that the Baltimore classification is not a phylogenetic scheme. So if they're in the same class of Baltimore, the same Baltimore class, doesn't necessarily mean that there's a shared ancestor. And the opposite is true, because viruses that have a shared ancestor can be in two different Baltimore classes. Okay? Oh, actually, I changed this around. Yeah. Oh, no. That's true, actually. I didn't update the slides for the French version. So, yeah, so this is an example. They're in the same class, but they don't have a common ancestor, as far as we can figure out. So, this website, which I absolutely recommend for everybody, the Viral Zone, it's got a combination of these two. So if you see that the main categories here, this is the Baltimore classification. Double-stranded DNA, single-stranded DNA, RNA, plus strand, minus strand, and then it's got these major phyla and of viruses that come from the ICTV classification. So it's trying to synthesize both of these things. Okay, we've got 4 minutes, so I will just go through these slides. So a lot of virologists spend time moaning about the ICTV classification, but it does allow us to come to some interesting conclusions about the origins of viruses and virus evolution. That's the whole point of phylogenetic classification. It's a model of the evolutionary pathways of different groups of biological entities. And it's the same for viruses as for species. So if you look at DNA viruses, and we try and think about viruses that infect bacteria, bacteriophages, and eukaryotic viruses. So up until the origin of the eukaryotic cell, 1.5 billion years ago, we just had bacteria. Right? So any viruses that existed must have been just infecting bacteria. So all of the eukaryote viruses, somehow they've emerged since the origin of eukaryotic cells. So where did they come from if they emerged? So for DNA viruses, if you look, pretty often you can see kind of like equivalence between some families of DNA phages, bacteriophages, and some eukaryotic viruses. Like Tectiviridae and Adenoviridae, they've got similar kind of structure of their genome, similar kind of similarities in the way the genome is replicated, and the capsid structure is also quite similar. So these are the characters that allow us to group these viruses together. And the same is true for the tailed phages, the caudoviricets, you know, bacteriophage T4, lambda, this kind of thing. There are some similarities between them and the herpes viruses. And so once again, these viruses are grouped together in the duplo-DNA viria phylum, I guess, or kingdom. So what's the conclusion? Probably that means that these eukaryote viruses, they're derived from an ancestor that was already infecting bacteria. And when these new types of cells emerged, some of these bacteriophages were able to adapt to the new host. Okay, so the evolutionary history of these DNA viruses goes back probably billions of years. Now, how about RNA viruses? Now, for a long time, well, one of the things you notice is that there are very, very few RNA viruses that infect bacteria. There's only one family called the Leviviricets, and until 10 years ago, there were no equivalents in eukaryotes. So there are some that infect plants, seem to be similar. But for the major classes of RNA viruses, there's basically no equivalent that infect prokaryotes. So what is the interpretation of this? Well, personally, I think it means that these virus families have all emerged since the origin of eukaryotes. It probably didn't exist as precursors before. Okay, so this is why I think at least the ICTV classification is useful, because it can help you get new biological insights into the origins and the phylogeny of these major virus classes. So I will end up there, and we see each other in 2 days, right? Thursday. Okay.